So I'd like to thank the organizers for giving me the opportunity to present the work that we've been doing on Friedreich ataxia and advancing gene therapy. So I'm trying to see. So Friedreich ataxia is the most common recessive ataxia affecting 1 in 30,000 people in the Caucasian population. It has an onset before the age of 25 years of age, and it's a progressive mixed spinocerebellar and sensory ataxia. So in addition to the neurological defects in the disease, it's also characterized by hypertrophic cardiomyopathy, which is thought to be a major cause of premature death in 60% of the patients. In addition, it also has an increased incidence in diabetes. The disease is a genetic disease. It's due to mutation in the frataxin gene. And as you can see here, or I'm not sure I want to point out, I'm not going to use that pointer, sorry. As you can see on this slide, the frataxin gene the normal frataxin gene has a GA repeat within the intron of between 735 lengths, but in the FA patient, there's a GA repeat expansion between 100 to about 2,000, which leads to transcriptional silencing and less of frataxin expression. And you can see here on Western blot analysis from patient material, where in the red stars you can see the two patients where you see very much decreased levels of frataxin. So one thing to remember is that in Friedreich ataxia there's a low level or a very low level of frataxin, but the frataxin that's there is a wild-type frataxin in most of the cases. So what do we know about the pathophysiology impact in both patients and models? So we know from very early on that iron homeostasis is affected in this disease. You can see on the left side purified mitochondria from yeast samples where frataxin has been deleted, and you can see iron accumulation. And on the right side you can see cardiac autopsies where you can see pointed out oops, I'll try again, no, but I won't be able to do that. So you can see the dark puncti, which corresponds to pearl staining for iron staining in cardiac autopsies. In addition, very early on also, we knew that there was iron sulfur cluster biosynthesis deficit, so these decrease of these activity of complex 1, 2, and 3 of the aconitase of the mitochondria, both in patients' cardiac biopsies and in yeast, and also there was an increase in oxidative stress, so a sensitivity to oxidative stress. And so this led to the idea that frataxin was involved, very closely involved in this vicious circle where you can have iron sulfur cluster biosynthesis defect, iron homeostasis, and oxidative stress. So what do we know about frataxin? It's a highly conserved protein from the bacterial all the way to the human. Has a ubiquitous expression. It's a mitochondrial protein, and it binds iron on its acidic bridge. There's been an extensive amount of work done on the function of frataxin, both in vivo and biochemically. And what we now know is that frataxin regulates de novo iron sulfur cluster biosynthesis. You can see this complex system within the mitochondria. So where frataxin in green is involved in regulating the cysteine desulfurase activity of this de novo cluster, where you need cysteine or sulfur coming from the cysteine desulfurase and iron coming in to be built on the ISCU protein. So iron sulfur cluster proteins are very important in cellular, in all cellular pathways. You know, iron sulfur clusters are these small inorganic prosthetic group, as you can see from this picture, they were very much involved both in mitochondria, and again, I'm sorry I can't point this out, but you can see on the top part of the mitochondria, we can see them in complex of the respiratory chain. You can see them in lipidic acid synthesis and heme biosynthesis and mitochondrial Krebs cycle. In addition, iron sulfur cluster proteins are extremely important for iron homeostasis within the cytosol, translation and ribosomal function, and in the nucleus, they're also very important for DNA metabolism. So you can imagine that loss of frataxin can lead to various amounts of dysmetabolism all throughout both the mitochondria, but also nuclear and cytosolic. So the approach that we've taken in the lab has been to try to understand the disease, and going into therapeutic approaches built in cell and mouse models. And as I said, the site of pathology in FA are several places. There's the cerebellum, and particularly the dentate nucleus. the large primary sensory neurons of the dorsal root ganglion, this leading to the spinal cerebellar ataxia, and the heart and the pancreas. And so we've built a number of different models using the conditional Crete conditional system. Here you can see the scheme of our crosses. So the constitutive knockout is embryonic lethal at 6.5 embryonic day. And so with the conditional models, we've developed different models, neuronal models, cardiac models, liver models. So today we'll concentrate on the cardiac model since this is the topic of the session. And so overall, these conditional mouse models reproduce important pathophysiological and biochemical features of the disease, with particularly the progressive cardiac hypertrophy, the deficiency in iron sulfur cluster activities, the mitochondrial iron accumulation. You can see here on this EM picture the dark puncti iron accumulation within the mitochondria. The neuronal models also reveal slowly progressive mixed cerebellar and sensory ataxia with progressive loss of proprioception. And so these models have really been able us to understand the pathophysiological pathway involved with loss of frataxin leading primary to iron sulfur cluster deficit, mitochondrial dysfunction and mitochondrial iron deposit, and then cellular dysfunction with increased oxidative stress. So there are different therapeutic approaches that have been done throughout the world for FA, and you can see here the pharmacological approach. Some of them are trying to increase frataxin expression, getting to very early on by looking at HCG inhibitors, for example, or nucleic acid-based approaches going around it's getting close to the GAA expansion mutation. Some other approaches have been trying to improve mitochondrial function, reduce oxidative stress, or modulate the frataxin metabolic pathways that are controlled by frataxin. So we decided to take a gene therapy approach, and the reason for that is because this is an autosomal recessive, postnatal onset, and slowly progressive. There's residual frataxin expression, so we imagine that there's not going to be an immune response too much around frataxin, and frataxin is a small protein. And based on a lot of different cell and mouse models, as well as healthy carriers of the disease, we hypothesized that very little frataxin is needed to probably have therapeutic effects. And so we've used an AAV-based approach. In this audience, I don't have to explain the advantages of using an AAV. The AAV that we use is the ARCH10 because this has strong cardiac and CNS tropism. And our therapeutic vector is human frataxin that actually has a HA tag from a strong CAG promoter. And so we evaluate this in post-symptomatic treatment in the cardiac mouse model. Here you can see the scheme of the cardiac mouse model where it develops biochemical features very early on at 4 weeks of age, and it develops progressive cardiomyopathy by 5 weeks of age. And here we're injecting at 7 weeks of age. And using this scheme, we were able to show a few years ago that intravenous injection of this AAV RH10 expressing human frataxin was able to prevent completely the severe mitochondrial cardiomyopathy. We were able to show that it improved the survival and the growth of the animals. There was rapid normalization of the left ventricle function and remodeling, and there was rapid stop of the cardiac fibrosis. And in addition, we were able to show that there was rapid rescue of the biochemical phenotype and the mitochondrial dysfunction, and we had a large therapeutic window because we actually were able to treat at different points of the disease. But one of the questions that we were asking ourselves is how to translate successfully these results into clinic, and really the proof of principle was we basically had 100% transduction of the cardiomyocytes. And so we really wanted to know what were the minimal percentage of cardiomyocytes to be treated to have rescue, because we know that in the human context we could never get 100% transduction. And so this is the work of Brahim Belbela, who is no longer in the lab, but he was a postdoctoral fellow in the lab and did an enormous amount of work on this. And so he administered decreasing dose of the vector in the MCK mouse model, and then looked at echocardiography, and then looked at survival, cardiac function, and a lot of other comorbidities. And you can see here on this slide, so we injected at 5 weeks of age, so this is already where the onset of the symptom is already there. And you can see the decreasing dose, and again, here you can just look at the error, the decreasing dose of the vector, and you can see here the left ventricle shortening fraction. You can see that with decreasing dose, we're not correcting this. And so the dose-dependent rescue of the cardiac function is clear. And we also had, I'm not going to show you all the data, but we also have hypertrophy and survival that is dose-dependent. Here is, if we look at the amount of frataxin, the VCN amount of frataxin protein, you can see that again, this is a dose dependence. You can see with high dose, we have high VCN, we're around 9 to 10 copies, where low dose, we're going down to the line. And again, the protein, the total amount of protein, of human frataxin protein levels also is correlated to the dose. On the right, we can see the correlation analysis between the shortening fraction, and here I'm showing you the VCN, but we actually have this for all the different markers in addition to the frataxin protein levels. And you can see very interestingly that we can see that there's a therapeutic threshold between 0.2 to 0.4 VCN, where above the 0.2 or above 0.4 VCN, we have a complete correction of the shortening fraction. Here, where you can see in the gray line is the normal shortening fraction, and where at 0.2 we have a quite strong correlation. And in addition, we were able to show that about 100 nanograms of frataxin per milligrams of protein was enough also to reach the therapeutic threshold. When we then looked at SDH activity, which is an iron sulfur cluster enzyme, we were able to show that we have also a dose response rescue of the mitochondrial iron sulfur cluster activity. And you can see here again, good thing I put a red arrow there. So look at the red arrow, we're going down decreasing doses, and we're looking here at enzymatic activity of SDH, and in blue is positive activity, and when you don't see any blue, you don't have activity. And so you can see, and on the bottom you can see the percentage. So this is, we actually quantified the percentage of the heart that was positive for SDH. And you can see that we have a decreasing amount of SDH activity depending on the dose. And this actually very nicely correlates also, which I'm not showing here, but with the amount of frataxin. So it completely correlates with decreasing amount of frataxin also. And so you can see here on the right, you can see the correlation that we have done. And if we look, for example, at VCN to heart surface SDH, you can see that, again, when we're around the 0.2 to 0.4 VCN, which I remind you was a full correction of the cardiomyopathy, you can see that we are between 50 and 60% of the cardiac surface that's being treated. And again, I'm not showing you this, but we were able also to show that with frataxin expression, if we have 50% or 60% of cardiac surface that is expressing frataxin, we have a full rescue of the cardiac function. And so this is actually very encouraging to be able to translate this into clinic, but there's still quite a bit of work to do. One of the questions we were asking is if we can try to find a predictive plasmatic biomarkers to be able to show that this was that we can see the correction also. Here I'm showing you a biomarker. It's called GDF15. It's a biomarker associated with ATF and mitochondrial stress response. And you can see here in the mouse model, non-treated mouse model, you can see that it's increasing with age. When we actually treat the mouse, here's 1 week after treatment. So 1 week after treatment, you can see in green that we rescue the plasmatic concentration of GDF15 compared to the red, where it's the untreated animals. And with this rescue, and so this is a very quick response because this is 1 week after treatment. And you can actually see we've done correlation again, and there's a nice correlation between the GDF plasmatic concentration and the heart surface that it's SDH positive. And again, so we believe that this might be a good biomarker, but of course we need to be able to figure out if this is going to be a good biomarker for the human disease. So at least in the mouse model, it's a good biomarker. So just very quickly at the end, I just want to finish with observations that we've made, and I think this is important. So while Brian was looking throughout the model, what he actually was able to see is that there was subclinical toxicity associated with frataxin overexpression, and I think this is really important to take into account. So you can see here on this picture, or I hope you can see, so this is on the left, you have frataxin immunohistochemistry in red, so you can see throughout the heart, different amount of frataxin, and in strong red is actually when there's quite a bit of overexpression. And then in blue, you see the SDH activity. And when we zoom in, I hope you can appreciate the fact that every time there's very high levels of frataxin, there's actually low levels of SDH activity. And so this really shows us that there's a toxicity effect of too much frataxin. We're actually impairing the enzymatic activity of SDH. And Brahim has done a lot of work on this and it has been able to correlate this with a lot of different mitochondrial dysfunction and iron homeostasis dysfunction. So clearly too much frataxin in the MCK animal here is toxic. So he then went on and tried to look to see what happens in wild-type mice. And I think this is important to show, and I don't know if the movies will be able to show, because I don't have any access to it, so if you can launch the movies, please. But, so what he's done is he's injected very high levels of vectors in wild-type animals and looked at the echocardiography. So, and what he was able to show, and then he looked over time and then sacrificed at 21 weeks of age. And so what he was able to show is that when he's injecting 5 times 10 to the 14 Vg per kilo, so this is very high dose, he was able to show that there was a decreased shortening fraction, although there was no overall cardiac output dysfunction at 21 weeks of age. So showing already in the wild-type context that high levels of frataxin can be toxic. This would be so much easier if I had a pointer, I'm sorry. So if I can get to the next slide. And again, he's done quite a bit of histological analysis. So again, this is in wild-type animals. And so you can see on the left side H&E staining, where you can see the fibrosis. You can also see the fibrosis with WGA staining. And then he's looked at co-staining between frataxin expression in red and SDH activity in blue in the middle. And then he also looked at COX activity and ADH activity. So these are different activities of mitochondria. And you can, I think, appreciate that at very high levels of frataxin expression in wild-type animals, he's seeing toxicity on at least on the mitochondrial function. And he's also seeing fibrosis. And so really showing that we need to be careful about the amount of frataxin that we're putting in. Of course, here this is artificial. We're in a system where we're putting huge amount of vectors. We have done more work on this, and this is currently under preparation, where we've increased the amount of frataxin expression by modulating the vector, by changing the vector, and where we can see that at low levels of vector with high expression of frataxin, we also get the same type of toxicity. So in conclusion, I think that we've had quite a bit of success showing proof of concept of gene therapy for the cardiomyopathy associated with FA, with rapid correction and reversal of the disease, and this has been published a few years ago. We just recently were able to show that a dose-response study demonstrated that about 50 to 60% of correction of the cardiac surface is sufficient to have full cardiac functional rescue, and so this is very interesting when we think about going into translating this into clinic. In the mouse, we've been able to see that GDF15 is a predictive biomarker for the animal model. So will this be a biomarker for the human, or could this be a biomarker possibly for the toxicity effect? And as I just showed you in the last few slides, that high levels of frataxin can be toxic or subtoxic. And so we need to very carefully design the therapeutic vector in order to be able to go into clinic. From all the different measurements that we've done, different vectors that we've constructed, which I didn't have time to show you here, is we've been able to see between 10 and 20-fold endogenous expression of frataxin appears to be safe, at least in the mouse. And so what I think we all believe in the field is that if we can get as close as to the endogenous level for expressing frataxin, this would be very important. And this is actually very difficult to do because different tissues have very much different levels of frataxin, and so we probably need to go with the endogenous promoter would be probably the best way to go about. So in terms of perspective, we're looking at the mechanism of toxicity. We believe that it's a toxicity directly on the iron sulfur cluster biosynthesis pathway, and we're of course designing new vectors to try to reach that low levels of frataxin. So with that, I'd like to acknowledge the people involved in this work. Actually, all the work I've shown you is the work of Brahim Belbela, who is a very talented postdoctoral fellow in the lab, who now no longer is in the lab, but is working for Adverum Therapeutics. And I'd like to thank the sponsors of this work, in particular FARA and AFAF, who've been very helpful throughout the years helping us developing this. And I'd like to thank you for your attention. I'll be happy to take questions.
So we are good in time, so please come to the microphones and ask questions. And also there is a seat over there.
Good morning. I have two questions. One question is, sorry, you want to go ahead first?
No, we're good.
Okay.
Go ahead.
If you were to the clinic in the human population, what would be the target age that you could see correction in the cardiac? That's my first question. And my second question is, when you look at the overexpression, the 10 to 20% or 20 to 20 full over endogenous, would it matter whether it's the number of cells or overexpression overall is okay? I guess my question is, would it matter whether it's the number of cells that overexpress the frataxin or it's just the overall overexpression?
Okay, so to answer to your first question, the target population in FA, this is actually a very difficult question to ask because we don't have any good predictive markers of knowing which FA patient is going to, so most of them develop a cardiomyopathy, but which one is going to actually go into cardiac decompensation and at what time is very hard to answer because we don't really have these biomarkers, and so there's a big effort in the field to try to answer that question. The second question on the overexpression. So there's two aspects. So the overexpression and the toxicity on overexpression, first of all, is cell autonomous, as the correction is cell autonomous. So there's no frataxin leaking out of the cells. It's really on the cell autonomous basis. So when we have overexpression within one cell, so we see the very strong frataxin levels, that's within one cell, and that's when we see the toxicity. Of course, when we're measuring levels of overexpression, we're taking the whole population of the cells, and so when we're saying 10 to 20-fold, this means 10 to 20-fold overall, but that means that some of the cardiomyocytes are going to be expressing much higher levels and some much lower levels because we know that it's not equivalent. So that's just an overall. So that's why I think that it's really important to try to go down to a vector that's expressing as close as possible to endogenous level. But as I said, this is very difficult because for the cardiac aspect, it's actually the cardiac express quite a high level of frataxin where, for example, the dorsal root ganglia neurons express very low levels of frataxin. So that's going to be hard to say. But I hope I answered your question. Yeah.
Great talk. What are your thoughts on looking into the immune response in terms of ELI spots to look at can you separate the FA versus the, because you're using a pretty high level of AAV, if it's more of a response that you're seeing in the heart?
Yeah, so I didn't have time to present all the work, so we actually don't believe it's an immune response. So at this point, it could be, some of it could be the immune response to the vector because we're using very high levels. But we've actually developed a vector that expresses very high levels of frataxin using low doses of vector, and we actually see the same toxicity. So we really believe that most of the toxic effect is based on the frataxin, not an immune response against the vector.
So with the small animal model, you essentially have much more expression in the liver than you do in the heart. So what are your thoughts if you scaled to a large animal model? So my group's done a lot of work with large animal gene transfer. We get more in the heart versus the liver. There are ways to do that in a large animal. What are your thoughts on that? Have you tried scaling up to large animal? And maybe there's not an exact model, but to look at the effects of FA with more expression in the heart versus the collateral to tease that out.
So I haven't personally tried because I'm not involved in large animal, but I think this is important to do. I mean, this is the reason why we wanted to try to figure out in the small animal model that has the disease, what's the percentage of cardiomyocytes that we need to obtain to be able to have a functional rescue. So I think that now what it really has to be done in large animal model is to find out doses that I'm quite confident if we can get to 50% of transduction in the large animal, then that probably, at least the data that we have in the mouse, suggests that this would be therapeutic.
We have a question there. Yeah.
Hi, great talk. Arvind Asogan, Duke University. I'm trying to connect the numbers that you put up. So based on the AAV copy numbers, you're looking at about 0.2 to 0.4. So that would suggest 20 to 40% of the cells are positive for the DNA. But then with the percentage of cells that are positive for frataxin expression, I think you were saying we want to be in the 50 to 60% range. So I'm wondering at what time point, and I might have missed this, you looked at the copy numbers per cell, because as we know, it kind of starts off really high and then starts tapering off.
Yep.
So trying to correlate those, and any help there?
So we actually presented this in the paper. The 0.2, 0.4 vector copy number per cell, actually, depending on how you calculate the vector number copies per cells, you can either have 0.2 to 0.4, or you can go to 0.5. So this is actually based on how we extract the DNA from the cells. And so we've actually been able to show that this copy number is about 0.5 actually, and not that low number. So that's just it's a technical problem. I probably should change my slides to bring that out.
Quick follow-up question. Have you looked at the different cell types? This is all predominantly in cardiomyocytes we're talking about, or endothelial cells, smooth muscle?
No, these are cardiomyocytes. So we've done the different markers to look at this, and this is really looking at transduction in cardiomyocytes.
Thanks.
All right, thank you very much.